So the kind of initial concept was a very simple one, and that was a kind of off-the-cuff comment by me at a meeting saying, well, what about getting stuff in and getting stuff out? And the idea here is that if you've got some kind of cell factory of any type, it could be bacterium, it could be yeast, it could be a kind of human cell line that you're using in some kind of IB process, then maybe you could improve the efficiency of that process by getting more useful things into the factory. So that involves crossing whatever membranes exist and cell envelope between the environment and the factory. So you may be able to manipulate those transporters that are involved in taking up your substrates that you can convert into your useful product. These may be native transporters that are already present in the factory, but you might want your factory to utilize a substrate that it might not normally use. So you might have to heterologously express a transporter from some other organism. Okay, so a kind of synthetic biology kind of angle there. Or perhaps you can take one of the native transporters and do some mutagenesis, either kind of random or site-directed, to alter the substrate specificity of that transporter so you can take up things that it wouldn't normally take up. So you can get more useful things into your factory, and it seemed to me that this might be a neglected area in kind of considering existing IB processes and maybe developing IB processes in the future. Another factor, if you're thinking about a kind of consolidated bioprocessing activity, is that you may have a very complex feedstock which needs breaking down into something useful before your factory can operate and use these kind of simple substrates to create your product. And in that case, you might want to express some enzymes from another organism in your factory and have them secreted outside the cell to break down your feedstock, which can then be, those simpler components can be taken up by the cell in order to produce your desired product. So there's all kinds of activity here around the membrane in shuffling things across and shuffling things in. In this case, it might be an enzyme that you're moving out to break down a feedstock, but it could be your product. It could be a recombinant protein which you want to excrete from your factory into the medium in order to facilitate your downstream processing. And that might involve either generating new transporters or finding signal sequences, etc., in order to facilitate that transfer. Once you've made your product, unfortunately, very often it seems that the product is toxic to your factory, and so it kills your cell. Okay, so one way to try and enhance the productivity and the lifetime of your factory would be to remove the toxic product from the site of toxicity. And so manipulating kind of efflux pumps to get your product away from the cytoplasm to enhance the activity and life span of your factory could be really important. So getting things out is important from that point of view, your product out. And then we shouldn't neglect the membrane itself, the proteins and the lipids that are in the membrane. We may be able to engineer them, change them, to make the membrane more tolerant of your toxic product, so we can increase, if you like, the robustness of the cell factory. So it seemed to me that all of these things were related to the membrane, that there may be opportunities there to kind of improve various IB processes as a result, maybe through minimizing toxicity or optimizing expression of transporters, exporting products to overcome yield restrictions, development of a kind of enabling technologies, so that ways, for example, of controlling gene expression, so you don't have uncontrolled expression of membrane proteins, which could cause problems with the stability of your factory. And all of those things could come together to improve existing and move forward, kind of embed them within new IB processes. So all of these things kind of involve a kind of, if you like, synthetic biology approach to things. So as part of the network, we'll also include the kind of sociological aspects of using synthetic biology and cell factories and genetically modified organisms in a kind of IB setting. So that was the idea. As Andrew has already said, we were successful. There are 13 BBSRC networks in this area, called affectionately known, I guess, as the NIBs. And as has been said, we're kind of down here in this cross-cutting trio of NIBs. And you can kind of see why we're there, because if you look at the other networks that are around here, if in any point in anything that they're interested in, for example, in bioprocessing or biocatalysis, there's a cell factory, then something is going to be crossing a membrane at some point. And so this provides us with lots of opportunities to work with these other networks through joint meetings and possibly through joint funding of projects. So as Andrew said, kind of bear things in mind when you're coming up with projects that they might fit within different NIBs, or they might fit between NIBs, or be of interest to multiple NIBs, and you can get joint support for your ideas through approaching several NIBs. Okay? As a group, we all have this kind of overriding goal or objective, if you like, and that is to foster the collaborations between academia, industry, policymakers, and non-governmental organisations to tackle some of the real research problems that affect the IB industry in the UK. So we need this industrial focus on the things that we're doing, and then translate that research through some of the mechanisms that Andrew has already told you about, through, for example, the IB Catalyst and other funding streams, to deliver some key benefits to the UK IB sector. So I introduced the management team earlier on, but once we were successful, we formed a management group and a kind of grandly named executive team. Again, the most important person in here is Jen. If you want anything doing, contact Jen, and we'll try and do our best to support your ideas. Okay, so we're relatively new. We managed to launch in June of this year. We got the website up and running, which most of you, well, all of you must have been on, otherwise you wouldn't have been here. Like I say, I want to try and get some more contributors to that website to kind of keep it current and fresh, so there's a reason for people to keep going there to check out what we're doing. There's a LinkedIn group. If you haven't joined up, please do. And there's a kind of Twitter feed, which we have a number of followers. Again, might be a good idea to kind of link into that and become one of our followers. And we've got about 340 members at the moment, okay, which is pretty good going. At the moment, the split is about 80% academia, 20% industry. I'd like to try and get the industry figures up a bit. So if you have contacts within industry who would be interested in our remit, then please encourage them to join, but not only join, to participate in the network, because we really want to get the kind of industry steer into the kind of thing that we want to do. We want to do research that's going to make a difference, rather than just interesting science. I don't mean just interesting science. We would like to combine very interesting science with industry relevant problems. Okay? So that industry steer is really important to us. This slide is simply to show that we haven't just got 300 members from the University of Sheffield and 40 from the University of York. We've got I've got a good range of institutions from around the UK and some in Europe. Okay, so we've got a good range of academics involved from various institutions, and we've got a good range of industry IB companies that have shown an interest as well. So we do have a good spread. We're not kind of focused in a particular institution. So what's our scope? This is really important. So what do we mean by industrial biotechnology and bioenergy? So here's a BBSRC definition. Okay, so we have to operate under this umbrella of what IBB is. So it's the use of biological resources for producing and processing materials, chemicals, and energy. Okay, and the biological resources could be kind of almost anything that's living. Okay. Some kind of cell factory there. So under that umbrella definition, then what is CBMNet involved in? Okay, So when you're thinking about applying for proof of concept funding, when you're thinking about the ideas that are going to come out in the next 24 hours for projects, when you might be suggesting themes for future events or meetings, keep within our remit, okay? So we've got to be within our projects must be directly membrane related, okay? We're interested in the functions, the roles, how we can manipulate membranes, okay? And very importantly, we want strong industrial support. We want genuine, real buy-in from the industry partners so that we can make a difference to the UK kind of IB activity, if you like. Okay, so can't really emphasize this too much, okay? The CBMNet as a kind of community is kind of here to serve, okay? So this is kind of my definition of what we're trying to do. People may disagree or argue about it, it's up to you. But I think we're a forum to foster collaborations between academia and industry to enhance the understanding of membrane-associated functions of cell factories, ultimately resulting in novel or improved production and processing of chemicals, materials, and energy. Okay. And the mechanisms we've got to try and achieve that kind of goal, if you like, are events such as this, so the kind of bigger meetings where we might discuss a large number of themes or several themes. We can have events where we have smaller meetings where there might be 12 or 6 of you want to get together, as I've said, to kind of scope out an idea, to meet some industrial partners, to see if there's anything that you can work on together. We can use the website to provide information, to let people contact each other, to use that as a communication to this group of members. And we've got some funding streams, which I shall come to in a second. And we hope with these kind of 3 major mechanisms to try and achieve several goals. So we want to try and widen participation. I think it was Jill who said we want to get people to take their expertise in one particular area and see if it can be applied somewhere in this IB world of membranes, okay? And try and think a little bit wider than just, you know, I work on gene expression and that's it, okay? In that sense, we're kind of developing a wider community, and building their interactions and transferring knowledge and building some expertise. We'd like, or I'd particularly like to encourage kind of early career scientists to get involved and get a bit of excitement going about industrial biotechnology that I perceive at an undergraduate level is not there when you talk to people about medicine and cancer and things, okay? There's no reason why it can't be as challenging and as exciting as kind of studying medicine. So kind of personal development from that point of view, trying to get the younger scientists of the future involved in this sector. We want to support competitive grant applications, so hopefully something will come out, some good ideas will come out of today's meetings and of future meetings that can get new collaborations together that will be able to apply for our proof of concept funding or directly to other funding streams that will be competitive and will be successful. Okay, so we're looking to build those future projects. Informing policy. I've already had to fill in several surveys for the BBSRC on behalf of the network. In your pack there, you will find there's another one, and I think Jen has some kind of bribery in order to get you to fill them in, to kind of get a network view of a particular area. So we're involved already in informing policy to some extent. And as I said, we do have some international members. We're basically a UK-based network, but we shouldn't be inward-looking. If there's expertise out there in the world that we need to get in to maximise our outputs, then we should draw them in. So they may not be able to get any hard cash from us, but they can certainly benefit from the interactions and they can act as partners in projects, and they can certainly come along to meetings and contribute to those things. So we should remember that international dimension. Okay, so in terms of funding, we've got 4 funding streams mainly, and the details I'm not going to go through. They're in the information pack, they're on the website. But we have these business interaction vouchers, which will give you £5,000 to work with an industrial collaborator. We have the proof of concept money, some of which will be available after this event. And we had the first round that closed a few weeks ago. As I said, we'd like to support kind of early career researchers, so we were happy to fund kind of bursaries for early career scientists to present their work at conferences that are relevant, and to maybe have exchange visits with industrial partners and things. And the other thing we're funding are kind of vacation studentships or scholarships for undergraduate students. So we'd like to get people to apply for vacation scholarships where they can have someone in their lab during the summer, an undergraduate, to work on a relevant IB project, in the hope that those young scientists will become engaged with the IB sector. So various schemes there. I think this is nearly my last slide. Is in terms of outcomes from the meeting, we were asked to kind of say what was success earlier on. For me, as a kind of, from a network viewpoint, I would like to think that this is the start point of building this community up into something that can really make a difference. We will initiate hopefully these new research collaborations between industry and academia, and generate some really innovative ideas based around the remit of the CBMNet. And as a kind of selfish sort of thing, I'd like to hopefully come away with a few names of volunteers for contributors to our website. Okay, and the final thing I will mention again is that as a result of this meeting, we hope to be able to fund some projects, if we can get some really good and innovative, risky, new ideas that are relevant to our remit, that are emerging from this event. So essentially, if you're here, you've got a chance of bidding, and the call will open on September the 12th and close on October the 24th. Okay, so that's a little carrot to kind of oil the neurons, if you like, when you're thinking about things. But I think the most important thing is to come up and identify what are the kind of key problems and what are these innovative solutions that we can try and overcome. And if you've got those ideas, then kind of some of this becomes irrelevant, because then you've got a good project. If you've got a good project, then there are many routes to funding, as we already see, through IPAs, through the IB Catalyst, through European funding, whatever it may be. Okay, so it's getting the ideas that's the important thing. Okay, right, thank you very much.